This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 94, recorded on August 6th, 2010. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, your weekly podcast about viruses. Joining me today from Western Massachusetts is Alan Dove.
Pleasure to be here.
How's it going, Alan?
Going well, keeping busy.
You know, I'm not sure you're in actually Western MA, more like Southwestern, maybe?
It's generally considered Western Massachusetts. Yeah. I'm right in the Springfield area, which is the big city in the western part of the state. But yeah, when people refer to Western Massachusetts, they mean everything west of Worcester.
Worcester. All right.
Yes.
Well, our next guest is from South Central Florida, and that would be Rich Condit.
North Central Florida.
Sorry.
Except I'm not there now. I'm in Texas.
Yeah. How is Texas?
Texas is Texas, you know. It's hot and flat. There's a few trees here. I'm just outside of Austin. I just got here yesterday, so.
Austin's a good town. I've heard Austin's good.
Yeah. Austin's beautiful. As a matter of fact, we're going to make a point while we're here of going and seeing the bats.
Yeah, that's the bats
that hang out under the bridge.
Yeah, at sunset they come flying out, right?
Yep.
Yeah, we had a picture of that on TWiV. Well, our special guest today is not far from you, Rich. Of course, it really is far, but in the sense of here to there, it's not far. That would be from the University of Utah, Dr. Ila Singh. Welcome to TWiV.
Thank you. It's a pleasure to be here.
Good to talk to you again. Ila was my colleague, I don't know, a couple of years ago here at Columbia.
That's right.
And left for greener pastures in Utah, but we miss you here.
I miss you guys too.
Ila is here to talk with us today about the novel retrovirus, the new retrovirus XMRV, and its involvement in prostate cancer and chronic fatigue syndrome. And before we do that, I like to just briefly review where people got to virology. I think I know most of your history because, of course, you were here at Columbia University Medical Center for a while. And before that, I know you were a postdoc with Pat Brown at Stanford, correct?
That's correct, yes.
And then you got your MD PhD with Ari Helenius at Yale?
That's right.
It's pretty good, I remember.
Yes.
That's as far back as I go.
That's plenty back enough.
And now you're at the Department of Pathology, correct? At the University.
Right.
Now, I remember once, years ago here, you were explaining the two different kinds of pathologists that exist, and you're one of them. So can you do that again for us?
Of course, yes. So there's two main branches of pathology, anatomic pathology and clinical pathology. Anatomic pathology is people who look mostly at tissues. So they're the people who, if you have surgery and if you have a tumor removed, then they're going to look at sections of the tumor and, at various immunostains and tell you whether it's a cancer, and if it's a cancer, what kind of cancer, etc. And they also are people who do autopsies and find out the cause of death or, things like that.
Okay.
Then there are clinical pathologists who are involved in developing and supervising the performance of all the other tests that happen when your blood is drawn or when you submit a sample of urine or stool or spit or, you name it. So I do the latter. So I'm a clinical pathologist.
Okay. I remember because I wanted to, if you remember, I wanted a collaborator and you said, no, you need an anatomic pathologist.
That's right.
Didn't even know there was more than one. Okay.
That's right.
So you're a clinical pathologist.
That's right. I mean, most of the time we work completely behind the scenes. So patients are often surprised that they have a pathologist's bill and they think that they never saw the pathologist, but actually the pathologist may have spent hours looking at their slides.
Right. Okay.
So, in your current job, are you what we call a physician scientist with some clinical responsibility and also a research lab?
That's right. That's right. I spend 80% of my time in my research lab, and then my clinical responsibility is in a lab called ARUP Laboratories, which is one of the largest university-owned reference labs in the country. And I help direct the hepatitis and retrovirus labs. So a lot of testing for HIV, other retroviruses, and
I have great respect for physician scientists because it's always struck me that that's like basically doing two jobs.
it
And
really is,
you
yes.
got a lot of balls in the air.
That's right. And the reason this current setup is a good one is that my clinical job is very focused and it's very closely related to my research job. That works out well. It's hard when the two are different.
Well, it seems like pathology is a field where you can actually pull that off, whereas a lot of other physician scientists are working in fields where the lab work is just going to be inherently very different from the clinical work.
That's right. That's right. And there's very little that you need to drop everything in the research lab and run to do to the clinical side when you're a pathologist. It's more so in anatomic pathologists who do frozen sections or something. But clinical pathology is a good field for some of your listeners who might be considering an MD PhD career that spans both sides.
Now, when you came here to Columbia from Stanford, you were working on murine leukemia viruses, right?
That's right. That's right.
But that's interesting because they're highly related to the virus we're going to talk about today. And
That's right.
I recall you giving seminars. So we had a virology group here at Columbia, and you used to come, and I heard you speak many times on Maloney. And then one day, suddenly, you were working on XMRV. To me, it seemed as though this came out of the blue. So can you tell us how you got into XMRV?
Right. So Joe DeRisi, who's at UCSF, who was involved in the original discovery of XMRV, talked to me about it when I was visiting him at UCSF. And this was just a little bit before the paper came out. And at that time, it looked like it was a murine retrovirus that was very related to the virus that we were working with. And they hadn't looked at any non-cancers. They just looked at prostate cancer. So it wasn't clear if this was a virus that was going to be present in all prostates or it was associated with prostate cancer specifically. There was also an association with mutations or variations in the RNA-seL gene. And RNA-seL gene, as you know, is involved in innate immunity. And there was some thinking about whether people with these variations may be more susceptible to XMRV. So, we thought that, so this was Steve Goff and I thought that, we were probably well suited to working with this virus because we had been working for so many years on the related other MLVs, the murine leukemia viruses, and this virus was very closely related. So it seemed that it would be worth finding out if it was truly associated with cancer or it was a virus that just liked to hang out on all prostates, and what the association with RNA-seL was and what was this virus doing. So we both started, working on this. In our own labs, we did different projects, and here we are.
And you have published a few papers looking at the involvement in prostate cancers, correct?
That's right. That's right.
And so what is the summation of that? What is the current state of our understanding in terms of prostate cancer?
Yeah, so what we did was we looked at about 233, if I remember correctly, cases of prostate cancer that were consecutive cases that came to Columbia University Medical Center for prostatectomy. And we looked at about 100 cases of benign prostates, that where people had their prostates removed for benign prostatic hyperplasia. And what we found was that the virus was present in over 1/4 of patients with prostate cancer and in only about 6% of patients without prostate cancer. And when we found the virus in cancer, it was almost always in the malignant prostatic epithelium. So, which is consistent with a retroviral theory of carcinogenesis, because in order for retrovirus to cause cancer, it needs to infect the cell from which the cancer arises. And until our study was done, Bob Silverman's previous study with the original report that reported XMRV had only seen the virus in stromal cells, very rare stromal cells, which is not consistent with how we understand retroviruses cause cancer. So this was kind of an interesting finding. And then we looked for the association between RNA-seL and XMRV, especially the association of those mutations in RNA-seL and XMRV, and we did not find anything, any association between the two. This was, again, different from what was reported previously. So, and that we've now done on a much larger subset of cases, and we don't find it. And now I think I've also heard Eric Klein, who is an author in the first study, which reported this association, saying that they don't see it anymore either. So, so that may have been a function of they're looking at a geographically restricted population where there might have been some linkage between that mutation, which we, in this very diverse population of New York City, we didn't see that.
So wait a minute. The first study that discovered the association between the RNase-L mutation and XMRV was screening. I mean, how was that done? Because it was looking for a genetic basis, right?
It's just looking for a single mutation, a point mutation that leads to an amino acid change from R to Q.
That would be correlated with, but they were looking for a correlation with prostate cancer, right?
They were looking, yes, so that was already known from a previous study, which had looked at hereditary prostate cancer and found that people who had prostate cancer in their families had this particular variant of RNase-L.
So the RNase-L correlation stands up.
No, surprising enough, no. So that correlation of RNase-L with prostate cancer was first reported in a very small study. Anytime a much larger population-wide study was done, that correlation does not hold up.
So in that case,
but there are only two such studies. So it's hard to say what is really going on there.
See, I'm trying to sort this out in my head because my original understanding was that you're looking for a correlation between some sort of genetic flaw and prostate cancer. You come up with the RNase-L mutation and an associated XMRV, and now those two are segregated. So I wonder whether, what's the truth? Is the reason the RNase-L thing came up is because you're actually selecting out people who were who had the XMRV, and the RNase-L mutation was closely associated? And it was the XMRV that led you to that discovery and not the RNase-L, now that the RNase-L is no longer a correlation. See what I mean?
I see what you mean, but really the people you should ask are the people who found that correlation, exactly how they picked their patients and how what was done. In trying to parse their paper very, very carefully, because our finding obviously did not agree with that, so we read that paper over and over, those aspects. We cannot tell how those patients were selected or what the process was. So all I can say is that our study involved, over 200 patients, so it was a much bigger study, and we didn't find any correlation. And now we have now done it on many more patients, and we still don't see that correlation.
So you see no correlation with the RNase L.
No correlation with the RNase L.
And where does that leave XMRV?
So all that means is that XMRV possibly can infect people with any kind of RNase L genotype. So maybe, so the implication would be that all of us are susceptible to XMRV regardless of what our RNase L makeup is.
Okay.
So have you continued to work on the prostate cancer XMRV connection?
That's right. Yes, a little bit.
Obviously, CFS may be taking your attention away from that, but I'm just wondering, where are we with that now? There have been a number of other studies. Some of them are positive, some of them are not. What has to be done, in your opinion, to sort that out?
Yeah, I think probably the biggest problem with XMRV, and I think this is equally true of prostate cancer and chronic fatigue syndrome, probably, is that I should address it more for prostate cancer because that's what we have most experience with. Is that the virus is present in samples in very small amounts. It really is, very hard to detect. Mm-hmm. And that's probably going to be the case with a lot of pathogens that are discovered now, is that, the ones that were present in abundant amounts are probably already known. Mm. And
the low-hanging fruit, right?
Yeah. So the problem really is that you need very sensitive tests. And at the same time, when you're working with a very sensitive test, you need it to be specific also. So these tests should be able to detect XMRV, but not detect other endogenous retroviruses or other mouse retroviruses. It can be a problem because a lot of people, when they section their tissues on microtomes, the same microtomes are often used to section mouse tissues. Mm. So Vincent, like the study that you were talking about when you needed a collaborator to do your mouse, those mouse tissues were probably sectioned on the same microtomes as human tissues are. Oh, so when you're looking for very tiny amounts of a virus, you want to make sure that you're not looking at mouse viruses. Sure. So we go through kind of a crazy routine, and we learned this the hard way. We make them change the knife, the blade, as well as bleach the microtome stage between cases. Sure. Makes the whole process much more expensive and tedious to do. So those are the sorts of issues. And then you really need to develop good tests, good sensitive tests. It's just not, not sufficient to show that something can detect something in a plasmid, template. It's hard to know if it's going to detect something in a matrix that's as complicated as blood or cellular DNA. So I think that's probably one of the biggest reasons for why people find different results.
So in your mind, is there pretty likely a correlation between XMRV and prostate cancer?
Yes, I think. I mean, I know that XMRV is present in prostate cancers. Okay, I'm pretty convinced about that. What I don't know is whether it's causing prostate cancer at all. So that that we are far away from showing that.
So I'm reminded of a previous show that we did where there were studies on the host range in culture of XMRV and the notion that it grew well on cells that came from prostates or prostate cancer cells.
That's right.
So what you're saying is that it could be that XMRV grows in prostate cancer but is not necessarily causative. That's a possibility, right?
Oh, absolutely. That,
And the association is not 100%. Of course, that could be a sensitivity issue as well, right?
That is correct. That is
so
correct.
It could be that you can't always detect it. It could be that it's not actually causative, but only associated, or it could be that it's causative, but only in, say, some fraction of prostate cancers. All those are possibilities. Is that right?
That's absolutely correct. Yes.
Okay.
So
and you would say that, I'm sorry. So you would say that there is conflicting literature on this, and you would attribute that to, say, sensitivity and specificity of assays used?
That's right. That's right. Yes.
Alan, you wanted to say something?
Yeah. So I keep wondering. I mean, the obvious explanation that occurs to me is that this virus is a bystander, that it's an opportunist, in, getting into these malignant cells in prostate cancer, and possibly there may be a similar story in chronic fatigue. And one way to kind of explore that would be to look for XMRV in other conditions where you might have particularly susceptible cells or particularly weakened immune systems. And I'm wondering if anybody's done that sort of study, like in transplant recipients or anything like that.
Not that I'm aware of. So we are doing a study where we have looked at prostate cancer in autopsy. We are looking for XMRV in an autopsy series. So we are looking at 75 male autopsies done over a period of a year and a half, every consecutive autopsy, and the same way for females. And that's still ongoing. There's a lot of analysis that needs to go on with these patients. And then we will actually be able to see if there's any correlation between presence of XMRV and transplant patients or any other immunosuppression, or patients who have other infections, sexually transmitted disease, and so on. So that's ongoing, and we should learn something from that, but it's too early to say.
Okay, so you're just screening everybody who comes across the slab then.
That's exactly right.
Multiple tissues as well, not just prostate or.
That's right.
Yeah.
That's right.
Well, that should be quite interesting. Yeah.
We wanted to know what other organs might be affected in XMRV infected individuals, and we wanted to know if females had XMRV. So these studies should answer some of those questions. We're not there yet.
The XMRV by sequence appears to be derived from a murine retrovirus.
Mm-hmm.
Do you have any sense of how that was introduced into humans at some point? Like, when did this happen and how?
No, I don't think anyone's done enough and to figure that out. The thinking is that it came into humans at some point, and then it has been transmitted from humans to humans.
Sure.
And it's not that every human infection is a result of a mouse encounter. But beyond that, I don't think we understand anything.
And even the prevalence in, say, healthy individuals, we really don't understand that level yet. Is that correct?
That's absolutely correct. I mean, in our case, there could be multiple explanations of why people with benign prostates have XMRV. It could be that our tests will pick up a certain number of false positives. So our test is not perfect, and the virus is not even there in these people, but we pick that up. The second possibility could be that these people have the virus but just haven't developed prostate cancer yet, and may develop prostate cancer in 10, 20 years. But these, with the samples that we have, are all completely de-identified, so there's no way of going back to these patients to see what happens to them. Right. And then the final possibility is that these are all the benign prostates were removed by a transurethral resection. And so that is an operation which just removes the tissue just surrounding the urethra. It's done because in elderly men, the prostate gets enlarged and they have difficulty urinating. So, but that part of the tissue, the part of the prostate that's around the urethra, is not the part that usually develops prostate cancer. Prostate cancer develops around the periphery of the gland. So it's possible that these people even may have prostate cancer in another part of the gland that's not yet discovered. So there are many reasons why you can say that there's a 6% positive, and we really don't know, and there's no good way right now of determining that.
A lot of stuff left to be done.
Yeah, it's amazing, right?
Yeah.
And we haven't even touched CFS yet.
That's right.
I had a quick look at your PNAS paper that described the association between XMRV and prostate cancer, and I was particularly struck by the fact that the percent positivity was higher when you looked by immunohistochemistry as opposed to PCR. So looking for protein expressed in cells by a microscopic technique. You stain with an antibody to the protein and look in the microscope as opposed to looking for DNA by amplification by PCR, because I'm conditioned to think that PCR is absolutely the most sensitive thing that you could possibly do. And you present compelling arguments for the notion that the immunohistochemistry is in fact more sensitive because it's only a few cells that are positive, and so by PCR the DNA that's present or the nucleic acid that's present is really pretty dilute.
That's right.
It's very, very interesting.
Yes. So sometimes the human eye is better than the most sophisticated test. And this is one of those examples, because you have this brown cell in a sea of blue cells, and the pathologist very quickly finds it under a microscope. It's easy to find.
The pictures were striking, and it really speaks to your arguments about selectivity and sensitivity of the assay. You got to have the right antibody, and you got to be doing the assay correctly.
That's right. And you got to be using the right tissues as well. A lot of people tend to think that they will just screen thousands of prostate cancers using tissue microarrays. And, we tried that and it doesn't work. And we have that on our website for people who are using our antibody. But I don't know how many people, then go back and say, okay, I'm going to, look at large sections, because the virus is really present in a few cells. So you really have to scan large sections, and looking at little tissue microarrays, in our hands at least, never worked.
And the antibody that you use, what's its origin?
So we cultured the virus in human cells, and then we harvested this virus and injected it into rabbits.
Okay, so it's made to complete virions.
That's correct. Yes.
Okay.
So early last year, the science paper came out showing a strong association of XMRV with patients with chronic fatigue, and you were already at Utah at the time, and so I assume that on hearing that, you decided to look into that as well.
Yes, we did. So that is another ongoing project. What we are doing is looking at 100 patients with chronic fatigue syndrome. We collected fresh samples from them, and we collected samples from 200 healthy volunteers. All of these people live in and around Salt Lake City. And we are analyzing and looking for XMRV using a battery of different tests. So using multiple PCR tests that look at different parts of the genome, doing RT-PCR, looking for virus, looking for the viral RNA. And we're using some of the same tests that the paper in Science reported. Namely the one where you take human plasma and you add it to LNCAP cells. LNCAP cells are a prostate cancer cell line that are particularly good at growing XMRV. And then you culture these cells for a while, and then you keep looking for evidence of XMRV infection in these cells by various means. So it's a whole bunch of tests. We're also doing some serological tests that we have developed. So we'll see what we find.
Are these different patients from the science study?
Yes, they're completely different patients from
Now,
the science.
how are you defining your CFS cases? Because that's been a bone of contention too, I gather.
Yeah, it's really hard. I mean, it's really hard. We have, different criteria that we have used to, so the Fukuda criteria, etc. And then the physicians have, categorized them into different severity groups, and we have people from very severe, moderately severe, and the most functional group. But these are very hard things to categorize. So one of the physicians who is involved in this study was explaining to us that sometimes you can call somebody very functional because they're holding a job, but then the nature of the disease is such that if they stress themselves out more, then they're likely to get more sick. So sometimes by our sort of ways that we measure function, we'll say, oh, this person is really functional. They actually hold a full-time job. But the real fact may be that that's all they do is they go to work, they work, and then they come back, and they're unable to do anything else. So these are very difficult categorizations, and it's a hard disease to just put a number on it, or put something quantitative that'll help us. So we're struggling, as is everyone else, to make some sense out of these.
What is the best material to use? So the original paper used blood or PBMCs. Are you doing the same? Are you doing more than that? We don't know where the virus is, right?
Right, that's exactly right. So we have no idea where the virus is. So we're just doing everything that we can. So we're looking at white cells in the blood. We're looking at plasma. We're looking at serum for antibodies. We're looking at we have a whole blood assay that we're using. So just using basically every fraction of peripheral blood that we can look at,
which is the easiest sample to get, I suppose.
It's the easiest sample to get from these patients, and really for chronic fatigue syndrome, it's hard to know what else might be the best tissue. For prostate cancer, it's obvious, right? You just use the prostate. So
it could be for CFS. It's a different tissue, right?
Yeah, I mean, it could be the brain, for example. I mean, who knows, right? Yeah.
So here again, my understanding is that there's been a lot of conflicting evidence or controversy in the literature about an association between CFS and XMRV, which again could be, the assays.
Yes.
What I've been looking for is for the individuals involved to swap samples. Has there been any of that?
Yes. So I can only tell you about what we are doing. And so Judy Mikovits, who's the main author of the original study that showed that XMRV was present in chronic fatigue syndrome, has been kind enough to make arrangements to give us samples, and we're sort of midway through getting samples from her. And this is how it's happening. So she gives the names of people who are positive for XMRV in their hands and people who are negative for XMRV in their hands to a phlebotomy service. The phlebotomy service then goes to these patients and collects the samples, and then they directly send the samples to us. So there's no question that the sample may get contaminated somehow in the Mikovits lab, which has XMRV growing there. So it's coming straight from the patient to us.
And are the samples coded when they get to you?
Yes, they're all coded. So we're completely blinded. We won't know which one is positive, which one is negative. Yeah. So we have so far, I think, gotten about 9 samples, and we'll probably get an equal number more. and then we'll start the analysis on those. So yes, so that is an ongoing process.
Excellent. I look forward to seeing that.
Yes.
Yeah.
I presume that this and many other studies are going to be needed before we really have a sense of where the virus is replicating, how it causes disease, and if it is associated with disease. Is that a reasonable statement to make?
That's absolutely reasonable to make.
Yeah. Because this is, from what I see, this is a growing field now. XMRV has had a meeting at Cold Spring Harbor. There's a meeting at NIH in September. So it's a brand new field. And probably you never thought that a murine virus would be clinically relevant in all those years.
That's right. That's absolutely correct. Yeah.
Now, you published a paper showing that XMRV is susceptible to some HIV antivirals.
That's right. Yes. So it is susceptible, probably most susceptible to a drug that inhibits HIV integrase called raltegravir. And then it is also susceptible to a couple of drugs that inhibit the reverse transcriptase enzyme. So AZT, which is a well-known anti-HIV drug, and then tenofovir disoprixyl fumarate, or TDF, which is the prodrug form of tenofovir, which inhibits RT. So those are particularly effective at inhibiting XMRV in cell culture. We haven't done any animal or human studies. So
this is an interesting situation because they're approved, obviously, for treating AIDS, and they can be used off-label.
Yes.
So, an XMRV positive patient could go to their physician and say, I want to have this. And so what would need to be done to have FDA approval for the use of these in treating, say, XMRV infection?
Yeah, so we are talking right now with Merck to start a clinical trial, which would be double-blind, placebo-controlled clinical trial in XMRV, in chronic fatigue syndrome patients. And the reason to do that was that it was brought to my attention that a lot of patients were already taking these drugs and were blogging about it on the web. So it just seemed wrong to not actually try to find out if by sort of scientific means if these drugs were effective or not. So we're in the very early stages of talking. It may not materialize in the form of a clinical trial, but that's what we are
I guess
engaged in.
I guess it's early in the story, so
Yes,
it's hard to generate the information you need to do such a trial because, again, we don't know where the virus is replicating and how it is pathogenic. But
that's right, and it's hard to even measure. How will you measure the outcome of your trial?
Yeah,
right.
Yeah.
Right, especially since CFS in particular is in many patients a relapsing, remitting sort of
That's
condition.
absolutely
So
right. That's absolutely
anything you give these people may appear to work in some of them.
right.
So the trial you described would be to see if individuals with chronic fatigue or their symptoms are ameliorated in any way by treatment. But,
it's not known yet how we would do it, but we're just in the process of just talking to see if ideally it would be great if we had a lab test which we could look at, if we could measure viral loads before and after.
Yeah, that's a good point because without that, it's hard to measure the efficacy of the treatment, right?
When if it's going to be hard to measure the virus in disease, then it's a hard thing to.
Yeah. The other approach is, of course, for testing, say, an HIV drug. You give it to a susceptible population, and then you see the fraction that are protected by it, right? Or a vaccine, even. But that can't really be done at this point with XMRV, I presume.
And even in the AIDS trials, I'm sure they measure viral load and stuff, right? So
yeah. That's
well,
exactly.
and you also have the confounding factor that those patients have HIV.
Yeah.
Yes, but also in the AIDS trials, I think the viral load came up much more, much later. And some of the early studies were, just done on the
Just done
basis
on
of symptoms and, T cell counts and, much more cruder assays than viral.
Assays run by pathologists, right? Not.
Yes.
That's a good point. No, you're right. Early on, we didn't understand much about viral loads, so that's a good point.
Well, my guess is you're not going to have any trouble getting people to enroll for this study. Sounds like there's the people who are afflicted by this are really desperate for some sort of treatment.
Yes, I think that, yeah, I sense that desperation when I see the blogs and people write to me all the time. But really, it's too early in the game for people to be, to even know what this virus is doing.
Yeah, and I think this is when people are talking about taking antiretrovirals, these are not benign compounds. It's a mighty big gun to aim at a target that we can't even identify yet.
That is correct.
I agree.
These drugs are not without their side effects.
Do you know how common CFS is in this country? What sort of numbers of patients we're talking about?
Yeah, people, throw around different numbers depending on what study they quote. Something like 0.5 to 2%. These are huge numbers.
A lot of patients. Okay.
It's a lot of patients. So
I was just thinking that when AIDS first surfaced in the 1980s, I just wonder if patients. I suspect not. I have a fuzzy memory. Maybe you remember, Ila, better, but was there the same type of patient response, or was it such an unusual and rare disease initially that you didn't have people saying, "I think I have this, treat me"? Do you know what I'm talking about? Do you recall the atmosphere at the time?
No, actually, I don't. So
I'm just wondering if this is a new phenomenon where from this point forward now a new disease or a disease that hasn't been sorted out, as soon as there's some suggestion of an infectious agent, will be clamoring for very, very quick solutions. And as you know, they never come quickly.
That's right. I also think that there's two things going on. There's the desire in the chronic fatigue syndrome patient group to be cured, of course, which is common with the people who had HIV. But in contrast, people with HIV, people took their disease seriously from day one. They knew that they had a disease. Whereas here, there are many people who are not even sure if chronic fatigue syndrome is an entity. So having a clear test, like, showing that there's a virus that is specific to this illness would mean more than just treatment. It would also validate for these patients that they have a real disease.
Right.
Okay, so there's that going on also in this.
Sure. Alan, did you have a last question before we do some email?
Yeah, one of the things that came up with the XMRV CFS link, and obviously that's still all up in the air, but already there have been discussions about CFS patients shouldn't give blood. And is that an appropriate precautionary reaction, or is that premature?
Well, it's probably an appropriate precautionary reaction. There's several countries that have already, asked people with CFS not to donate. So, Australia, Canada have already done that. And considering that there are not that many CFS people who are going out to donate, it's probably not a huge risk. But it would probably not be a bad precaution to have until we know a little bit more about the disease.
Okay, kind of like saying people who visited Britain for more than a month or whatever in the wake of
mad cow
BSE. Yeah.
BSE, yeah.
Right.
They were asked not to give blood either, right?
Right.
That's right. That's right.
Right, but before it was really known what risks that might entail, just the reflexive reaction. Right.
Yeah. I think
it seems appropriate to me too.
I think you have to balance it always, right?
Right.
As Eric Delwart said, he said there are a lot of on TWiV a few weeks ago. He said there are a lot of circoviruses in blood, and if you threw out every pint which was circovirus positive, you wouldn't have a blood supply any longer. So in that case, knowing that you've been giving it to people for years, you make a judgment, I suppose.
Right.
But
whereas in this case, how many CFS patients are actually giving blood? It's probably a minority to begin with, and you're not going to destroy the blood supply that way.
Okay, I'd like to do a few emails. We usually do email on TWiV, and today they're all with respect to XMRV and mainly CFS. So we'll see how many we can get through. The first one's from Kate.
I think it would be interesting if you highlighted how you got these emails, because I was astonished.
I had told the CEO of the CFIDS Association of America, the CEO who's been corresponding with me for a while, and the news spread. And I've had, 50 or 60 emails with multiple questions, so we won't get through them all, but we'll save them and have Dr. Singh back at a future date. I'm sure you'd love to do that. The first one is from Kate, who writes, Is it possible that disease stage makes a difference in detection of XMRV, and have any XMRV studies tested patients for the virus at several different points over a period of time?
Yes, it's quite possible that the disease state may have an effect. And no, I'm not aware of anybody who's tested multiple samples at different times. But, there's precedence for that for other diseases too. Like if you're looking for people who are bleeding into their intestines, you collect for occult blood and stool, and you collect that over several days in order to be able to detect that. So yes, if XMRV varies very widely at different times, then it's going to be hard to pick it up.
Right.
In your PNAS paper, you found, did you not, a stronger association with high-grade tumors in prostate cancer?
That's correct.
So that would, does that qualify as a disease stage, perhaps making a difference?
Yes, it could. Yeah, it could. Or it could be that the kinds of tumors XMRV leads to are the more aggressive kinds. I mean, I just don't know. We don't know enough.
Right. Okay.
Okay, the next ones are from Court, who sent in 1, 2, 3, 4, 5, 6 questions, but you've actually answered 5 of them, I believe.
Great.
So I think let's see, this one is interesting. Since the science paper was published, at least 6 papers have been published that were unable to find XMRV, and other studies have reportedly failed. Have there been other retroviruses or virus studies in which the community initially experienced a similar degree of difficulty validating, but later did validate?
I'm not aware of that.
Yeah. I think, I don't know the whole history, but the association of papillomaviruses with cervical carcinoma took quite a while to validate, I think. And I don't know what the initial reaction was, but I think it took, really a long time to really nail that down completely, having to do, among other things, with issues that we've already talked about, and that is, how do you detect it? What sort of samples do you take, and what are the assays that you use?
I think you're right about that, and I think even HIV was difficult initially.
There are some people who still deny it, right?
That's right. So I think that the bottom line is that scientists and clinicians want to be certain, and it takes a long time to get that certainty.
Yeah.
Well,
I think a lot of the. Oh, go ahead. All right.
No, you go ahead this time.
All right. I'll go this time. I think a lot of the public discussion and why it's gotten so loud is that most of the public is not accustomed to evaluating early stage science, and they don't understand how vague things are at this point.
Yeah, and I think I was going to comment in a similar vein that this is a great time to be doing this, to watch this process in action. As I look through all of the reports coming out on XMRV associated with CFS or prostate cancer, what occurs to me is something we've said time and time again, and that is that science is self-correcting, because it has to be validated by several people in several different laboratories, and it will all come out in the wash. And it's important to watch that happen and be conscious of it. It's great.
And also not to leap to judgments before that process has happened.
That's right.
Okay, the next ones are from Flower and C. Let's see, she or she has 5. Do you think XMRV is the result of vaccines, either human or pet, contaminated with an animal retrovirus?
There's no evidence for that so far.
Could it have dropped into or piggybacked onto a herpes virus like HHV6 or EBV, which allowed it to be spread casually?
Again, no evidence. Yeah.
How can cluster outbreaks be explained?
Of CFS.
Of CFS, sorry. Thank you for keeping me honest there.
Well, I mean, I guess, one way would be some infectious agent.
Yeah, sure.
Another way is if there's a psychosomatic component, which, by the way, for our listeners' benefit, is not the same as saying it's all in your head. Right. There are genuine psychosomatic conditions that occur in clusters, and the one that I always refer to is seasickness. It's a psychosomatic condition. If you think about it, there is nothing organically wrong with somebody suffering from seasickness, but seeing somebody else suffering from it increases your chances of suffering from it.
And seasickness is real.
It is absolutely 100% real. I can guarantee you that, and it can actually be fatal. So yeah, there are people who've died of dehydration or come close to death from dehydration from vomiting so much from seasickness. So I think that's the kind of analogy I would point to, is you can have clusters of just about anything.
So the implication of this question is that there are cluster outbreaks of CFS. Is that correct?
Yes, I think that is a known phenomena. So there have been clustered outbreaks of CFS. And some of the patients that were included in Judy Mikovits's study from Nevada, I think a fourth of them were part of an outbreak that occurred in the mid-'80s in the Incline Village area, or Incline Valley area in Nevada.
Wow.
Okay, let's see. Gay writes, he or she also has multiple. XMRV growth is stimulated by androgen. If XMRV should be shown to be the cause or a cofactor in CFS, why are most, like 4-to-1 CFS patients, women? And related to that, the macaque monkey studies showed that XMRV has a tropism for prostate tissue. Do we know what tissues it prefers in women?
No, so that's something that I'm hoping our autopsy series in women can shed some light on. We don't know that yet, and it's important to remember that these steroid response elements are not entirely specific. So it's not you can't just say that it only responds to androgen. It may respond also to estrogens and other hormones, but it's possible that it responds to them at different levels. So no one has looked at that, and someone needs to do it. But those experiments haven't been done. Steroid response elements can be more promiscuous than
right. One is from Kate, who writes, my question for the brilliant Dr. Singh is, if XMRV is it, then when would drugs be available to treat us if everything went to plan? Months, years, or decades?
So, we know that you can prevent XMRV replication in cells. We know that today. But, you would need to do human studies, as we were talking about before we, it was considered useful for anything. And of course, even before that, we need to know if XMRV is actually causing any of these illnesses.
Not months.
It would take a long time.
Yeah,
years,
at least.
Years.
Yeah. I mean, the good thing going is that these drugs, at least in the doses used for HIV, are the safety studies have already been done.
Right.
So if the same doses will work, then at least that long process can be avoided.
Okay.
But who knows what doses will really need to be used.
The next one's from Jackie, who writes, I was very encouraged to hear that you're engaged in a major study of XMRV and CFS with Dr. Bateman and Dr. Light. I guess it's public knowledge that you're doing this, Ila, right?
It is now.
Well, this person knew already. I didn't tell her, because I didn't know. Let's see. The first one is a time frame. If strong association only, what kind of studies do you think we might need to discover whether there is causation?
Yeah, so that's going to be hard. Animal models. For prostate cancer, we are doing a study on tumors where we are looking for integration sites. So XMRV is a gamma retrovirus, and other gamma retroviruses cause cancer by their genome inserting into the host chromosome in a way that allows the host cell to replicate faster. So in other words, the virus inserts its own genetic material next to a gene that is somehow involved in promoting growth. So the experiments we are doing is looking at where these viral insertions are in prostate cancer. And we're looking for two things in particular. We're looking to see if these insertions are near genes that promote growth. And we're looking to see if these insertions are clonal, which means that does every cell in the tumor have the same particular insertion? If we find those two things, then chances are very good that XMRV could be called a cancer-causing virus. But we're not there yet. It has been an extraordinarily difficult project. We're doing it in collaboration with Rick Bushman at Penn, who is one of the world's best experts on looking at integration of retroviruses into cells. And yet, what has made this project incredibly difficult is the presence of the virus in tumors is not, it's not there in huge amounts. That's made everything very difficult to do. So those are the kinds of things we're doing.
So you need a plausible mechanism.
Yes.
In order to really solidify the causation.
Correct.
Interesting.
One more from Jackie, which we've addressed, but I like the way she put this. It's very good. Is the reservoir for XMRV likely to be the most practical location from which to harvest XMRV samples and develop tests for use in a clinical setting, i.e. clearly saliva or blood tests are more practical, but what if the main reservoir is the brain or the nervous system?
Yes.
It's a problem, yeah, as we discussed, right?
Right. Oh.
So Jackie writes, the UK may seem a world away to you all at this time, but I can assure you that CFS-labeled patients over here are enthusiastically following all promising scientific developments in this field worldwide and are very grateful to you and your colleagues for undertaking this groundbreaking research and for your scientific curiosity. That's for you, Ila.
Thank you.
Writing from
Thank
UK.
you.
Rebecca writes, in treating XMRV, some of us are considering getting stem cell treatments. Do you think heart would hurt the new stem cells we get?
I'm sorry, I'm still picking my jaw up off the floor.
Stem cell treatments.
If Rebecca is listening, please do not get a stem cell treatment for this. That's horrifying.
What does Ila think?
Oh, I don't know what she means really. I mean, what stem cell treatment?
This would have to be something totally off-label.
Well, for HIV, you can get stem cells where they put in inhibitors of virus and CCR5. They take out your stem cells, modify them, and put them back.
Right.
But here we don't know. We don't have any way to inhibit XMRV in this way.
Yeah, and with HIV, we don't even know what the long-term prognosis of those patients is.
Right.
That is
yeah, don't do that.
A horrible idea. And whether heart would hurt the new stem cells, I have no idea. I think the new stem cells might very well hurt you.
I think Ila would know that. I know that for various transplants, you don't get any, like for liver transplants after Hep C, you don't get any treatment for Hep C or HIV for months after the transplant, right? Because that's never been clinically tested in that sort of situation. Am I correct there, Ila?
That's right. But eventually for Hep C liver transplants, the new liver does get infected.
Right.
But, it buys time. It buys you, 5-10 years.
Right.
So therefore it's worth doing still. I don't know what a stem cell transplant, what the role of that is in chronic fatigue syndrome. I just don't know.
Okay. Terry writes, I'm wondering if Dr. Singh could comment if XMRV may be present in cancers other than prostate cancer. I'm wondering about cervical cancer in women. Is there a chance it could act as a trigger and interact with a person's genes and HPV to create cervical tumors?
Yeah, so that's a study that's ongoing right now in two ways. One is the autopsy study that I talked to you about, where we're looking at tissues in women. The other thing we're doing is we've gotten our hands on about 300 cervical fluid samples from 300 women, and we are looking at to see if we can detect XMRV in those samples. It's still not going to get to the question of whether it causes cervical cancer or not, but seeing if it's there in those tissues might be a start, and then we might be able to then look at a series of cancers of whatever organ we find XMRV in first. We haven't gotten that far yet.
Right.
Just as a little more background, I think it's quite well established that the human papillomavirus is a causative agent of, or the causative agent of cervical carcinoma. The association is virtually 100%, and the mechanisms are quite well worked out. However, not all individuals who are infected with or even have integrated HPV genomes develop cervical carcinoma. So there's only a small percentage, and so there's the idea has always been around that there may be cofactors. And so it seems to me that the search here is for the possibility that XMRV could be a cofactor in development of cervical carcinoma. Does that seem a reasonable summary of that?
That seems a perfect summary of that. And I think it's also important to note that HTLV1, which is another human retrovirus, causes T-cell lymphoma, T-cell leukemia. But it causes those cancers only in 5% of people who are infected with HTLV1. The other 95% don't seem to get it. And again, we don't understand why that happens.
So there could be possible cofactors there as well.
It could be possible cofactors there too, yeah.
Okay, let's do two more. One from Eric. In case XMRV causes CFS, to what degree do you expect people with CFS to recover given the right treatment? Not the treatment available now, but any future one. Meaning
that if XMRV is causing it, would antiretrovirals help?
Yeah. Or something specific
I think
for XMRV.
Yeah, I think the question is if it turns out that XMRV causes CFS and you wiped out the XMRV, would you cure CFS?
Okay. I don't know.
Yeah.
I really don't know.
That's fine. It's important to let...
I don't know is a good answer.
Yeah.
Yes.
It's from an expert, because we don't know how it causes disease. So.
Yeah.
And the last one is from Michael. Would it be expected that XMRV would have a similar disease effect as related viruses in mice, such as SFFV and MULV? If that's true, could we expect that the reason we see various outbreaks and susceptibility to XMRV be the same as in mice? And he sends a link to a Journal of Virology article in mice. SFFV, spleen focus forming virus vulnerability, can be either genetic or affected by a co-infection. If XMRV also turns out to be facilitated by a co-infection, could that cause the observed clusters?
Sure. I mean, any and all of that is possible. Yes.
I guess we haven't talked about it at all, but another possibility is that XMRV plus something else may be a causative factor, right?
Oh, of course. Yes. Yes, I think we've covered all possibilities that XMRV is just, doing nothing. It's just just present, or it could be causative, or it could be, just a cofactor.
Right.
There's still a lot to learn.
All right. Well, that's most of them, I think. And a few left we'll save for next time. I know you have to get to a seminar. Would you like to sign off now, Ila?
Sounds good.
We can
Vincent.
let you go. Thanks so much for joining us.
It was a pleasure talking to all of
Thank
you.
you.
Thank you very much.
Ila
Thank you,
Singh.
Richard, and thank you, Alan.
No
and
problem.
Thanks, Vincent.
You're welcome. Bye-bye.
Bye.
Let's do some picks of the week, gentlemen.
Ready?
Let's start with Alan.
My pick of the week is going to sound totally boring. It's the Federal Register.
That was my reaction. Sounded boring to me.
Yes. And a few years ago, I signed up to receive the table of contents of this thing. Now, if you're not familiar with it, the Federal Register is where executive branch agencies publish everything they're doing. And new rules that they're going to implement and all of this sort of thing. And so as a science journalist, I was interested in following science policy as it was being formed. And I subscribed to the table of contents for the Register for about a year. And the problem is this thing comes out every business day, and it takes half an hour to grind through the table of contents. Wow. It's just the volume coming out of these departments is astonishing. So as part of this whole new transparency push, there are a lot of government agencies that are putting new data online for the first time. But what they've done with the Federal Register, and this just, at least I just came across this recently, it was already all being put out there. The problem was you could hardly ever find what you were looking for because it was so much noise. To somebody else, it's fascinating to read about regulation of the telephone system, but not so much to me. And whereas to me, I'm fascinated in what's going on at the NIH, but maybe not so much to somebody who works at Verizon. Now, what you can do with the new version of the Register is subscribe to just a particular subsection. So you can pick the science and technology related stuff, and you can even narrow it down further. You can subscribe. They have RSS feeds. You can subscribe to the RSS feed for just featured articles in science and technology, which it looks like I've been doing this for a couple of weeks, and it looks like maybe 3 or 4 things a week come up in that. And so now it's actually a very straightforward thing to monitor what's going on and keep up with this. And, for people who say they don't care, I bet you care when these regulations actually come out. So it's kind of useful to be able to see them in the process. And you also have the opportunity to comment.
Yeah, this is amazing.
It is pretty amazing.
The safety, inspecting the low-pressure flex hoses of Boeing 767s.
Yes.
Changes to treatments for cherries from Australia and a radiation dose for fruit fly. Amazing stuff that we have no clue about.
Yeah.
Right. This is great.
Yeah, as Alan says, the volume is incredible.
Wow.
I like the tabs up on top of the homepage. Money.
Yes.
Money.
Right?
Environment, world, science.
Right.
Yes.
That's very cool. That's nice.
Terrific. I've heard of the Federal Register. I had no idea what it is, and I certainly didn't know this existed. This is terrific.
Yeah, anytime a federal agency is going to regulate something, they have to let everybody know in advance in the Register, and they usually have to have a public comment period. And then they have to answer those comments a certain amount of time later. So, you can participate in this process. A lot of people think that this is all just the government does stuff and they have no power over it. You can actually participate in this, but the problem has been drowning from trying to drink from a fire hose, and they've very neatly solved that. You can tailor the information feed to just the areas that interest you now.
Yeah, that's nice.
Cool.
Thank you for that. That's a good one. Rich.
Okay, I got another local one. This is the Florida Museum of Natural History Butterfly Rainforest. This is a branch of the Florida Museum of Natural History here in Gainesville. And I will offer the first 10 people who come to me mentioning TWiV free admission to this. Okay, this is a destination, I'll tell you. The Florida Museum of Natural History has made a real effort to make its mark relative to butterflies. And they now have the second largest collection of butterfly specimens in the world. They've got like over 4 million specimens and a space dedicated to a collection that can be expanded up to 20 million specimens. The only larger collection in the world is in London. And beyond that, they have built what I would call a vivarium, which is a 6,400 square foot exhibit that's really nicely landscaped. It's got waterfalls and ponds and stuff and all sorts of tropical plants. And at any given time, 60 to 80 species of butterflies flying around, several hundred at a time. And they only live a few days, so they release new butterflies that they actually purchase from butterfly farms around the world several times a week. We just went here we went there with my daughter and granddaughter last week. It's the second or third time I've been there. And you walk in and it's amazing. It's almost like being underwater with these butterflies just fluttering all over the place. Very colorful, all different species, and people walking around to give you tours. So this is a destination. Come to Gainesville and see the butterfly.
I didn't get to see this on my trip.
Next time, Vincent. Next time.
This looks really cool.
I'll probably be by this year sometime, and we'll go. That'll be neat. That's beautiful. There was a temporary exhibit here at the Museum of Natural History years ago, a butterfly. It was very small, and it was amazing.
The collection itself is amazing. They have a wall outside the vivarium that they have a special name for. I forget what it is, where they have a bunch of butterflies, parts of the collection mounted on the wall, organized in, I think, a geographic pattern. And they're astonishing. They're gorgeous.
Very nice. Check it out if you're in Gainesville, and mention TWiV to Rich and get free entrance. Excellent. Well, my pick.
I'll come with you.
You go as well, sure. You could go over and over, right?
Sure.
My pick is JOVE, the Journal of Visualized Experiments. This is a very interesting site. It is a peer-reviewed, PubMed-indexed journal devoted to the publication of biological research in a video format. It's basically people showing you how to do experiments on video. Here we have on the front page today, transurethral induction of mouse urinary tract infection. We have generation of recombinant influenza virus from plasmid DNA.
I was looking at that one. That looks great.
So it's people who video themselves in the lab doing techniques, and so you can see how to do it. It's more than just a protocol on a piece of paper. Pretty neat idea. Not a lot of virology, but all of science, biology here, neuroscience, infectious diseases, immunology, medicine. It's pretty neat. So
and it serves a real purpose, because as anybody who has trained in science knows, you can't figure out how to do this stuff just by reading the literature.
That's right.
You have to have somebody show you.
Yeah.
Yes. I've blogged about these guys. I blogged about these guys when they were first getting started a couple of years ago. I met the founders at a meeting that I went to, and then I just thought this was a really, really cool idea. And it is.
Yeah, it's great. I have always wanted to make videos of virology techniques, plaque assays, growing viruses, and put them here among other places. I just haven't done it, but this is a good idea.
Well, Adolfo Garcia Sastre has already.
He has, yeah. You have to. I mean, you can hire them to tape you. It costs a good amount of money. They'll come to your lab, or you could videotape yourself, in which case it's a little harder. You need to get someone to do it, but one of these days I may do that. But
this flu thing is amazing. They've got the transfection, harvesting the cells, harvesting the eggs, an HA assay.
It's very good.
I'm going to have to look at this whole
Check
thing.
it out.
I might use this for my classes.
The cool thing is that these are in PubMed. They're refereed. People look at them and say it makes sense or it doesn't make sense, and I guess you have to do revisions and retape it.
Right.
It's pretty cool.
Yeah, they're peer-reviewed videos.
Very neat idea. And that is my pick of the week, and that will do it for another TWiV. You can check us out at iTunes, the Zune Marketplace, and if you do, you can subscribe and get our updates for free, of course. You can leave comments there if you are new at the iTunes store. If you go and leave a comment and say what you think, it helps keep us on the front page of the Medicine Podcast Directory. You can also listen to TWiV with Stitcher Radio, which is a free app for your smartphone, and it will stream the episodes to your device. You could check that out at bit.ly/StitchTWiV or just search for Stitcher Radio on your smartphone. TWiV's part of microbeworld.org, sciencepodcasters.org, and promednetwork.com, where you can find other high-quality science podcasts. Don't forget to buy your TWiV merchandise at cafepress.com/TWiV. And if you don't like what's offered, let us know. We'll customize it for you. I thought Ila was great. I really like her.
Yeah.
Yeah, it was good fun.
And we will have her back at some point in the future. Maybe when her study's done. It sounds like that's going to be a while getting that done.
Right.
Yeah, it's going to be a long time.
Ila's at the Department of Pathology, University of Utah, Salt Lake City, and we'll put a link to her website. Alan is at alandove.com. Good to have you again.
Good to be back.
And Rich is, of course, where else? At the University of Florida, Gainesville, home of the Fighting Gators. Thanks, Rich.
Great to be here always.
When you going home?
Let's see. We fly home on Tuesday, and then I'm done traveling for a while. Then it's back to the pits.
Back to the pits.
Go back and work.
Yeah,
Get something done.
Well, you're going to travel a lot and visit your kids.
It's been a great summer. Well, yeah, we kind of made a commitment, my wife and I, to visit the grandchildren as often as we can, and they're scattered all over the place. So that's a lot of traveling. That's okay. You go to Seattle for the weekend,
Not bad.
Make the commitment.
Bad. Gentlemen will be back next week, both of you, I believe. I have you on the schedule.
Yes.
Yep.
And I think that will be a regular TWiV with stories. We'll catch up.
Okay.
You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.